That brings us to chapter 10, where we will discuss the skin's endogenous antioxidant network. And again, what we mean by endogenous are things that are inherently present in the skin. And this doesn't mean that they're always there. We've somehow acquired them through our diet. They may have been constructed of smaller things that we obtained in our diet, but many times the exact antioxidant molecule is transported to the skin from the gastrointestinal tract. A good example of that would be the carotenoids, also vitamin E, and we'll see several other examples in this chapter. So we'll briefly discuss defense systems against free radical damage, and then get into the endogenous antioxidants in skin. And of these endogenous antioxidants, there are several that we really want to focus on. So usually when we talk about antioxidants in the skin, we talk about small molecule antioxidants, and those are things like ascorbic acid, vitamin E, which tocopherols fall into this class of vitamin E. Glutathione would be another small molecule, and also the carotenoids. In addition to that, we'll also talk about the enzymatic antioxidants that are present in skin, and these consist of two of the principal ones that we really want to be concerned with are superoxide dismutases and also catalase. So this basically gives you a diagram of how defense systems operate and how they function against oxidative damage. You can see in this chart, you start off at the top with sources of free radical damage. Just some examples include hydrogen peroxide, solar radiation or light, smoke, metal, and oftentimes pharmaceutical compounds. We can have several different types of antioxidant strategies that are taking place. We can have preventive antioxidants. These essentially suppress radical formation. If these operate properly, you will not reach the step where you produce free radicals. However, if the preventive antioxidant system becomes overwhelmed, then you will produce free radicals, and then you go to the next step, and you can have free radical scavenging antioxidants, actually just radical scavenging antioxidants, which can essentially suppress this what they call chain initiation, because there's like a chain of events that occur. One molecule becomes damaged, that molecule damages another one, so you have the formation of a free radical. That free radical can react and damage another molecule, and then it becomes a free radical, and then that molecule will then react with and form another free radical with another molecule. So you have like a chain of events that occurs, and you can have radical scavenging antioxidants that actually suppress that, or you can have ones that basically break the chain propagation, and they basically stop the reaction right in its place. We usually, when we talk about free radical damage, the molecules that are most of interest to us, that we're most concerned about, are lipids, proteins, and of course DNA. And DNA, if you damage lipids and proteins, that can be detrimental to the cellular homeostasis. But if you damage DNA, that's where a lot of problems come in, especially because at that point you could be changing the genetic makeup within the cell, and this could eventually lead to carcinomas and things of that nature. So if the radical scavenging antioxidants do not do their job, you'll end up going to this next step where you have chain oxidation. And then at that point, this is where you can have damage taking place. And, before you arrive to diseases, cancer, or some type of aging, either chronological or photoaging in the case of skin, you can have these repair and de novo processes taking place that can actually repair, damaged species in the skin. So, that's the goal would be to have, one of these lines of defenses to actually stop the damage from occurring or repairing it before you reach the most detrimental state, which is the diseases, cancer, and aging. When we talk about endogenous antioxidants in skin, again, we have antioxidant enzymes, which I mentioned. The two that we'll be most concerned about for this chapter will be superoxide dismutase, and there's actually several different forms of that. And then we also have catalase, and we'll discuss those in detail in terms of what type of molecules they neutralize. You also have things like glutathione peroxidase, NADPH quinone reductase, and the thioredoxin system. These are also very important, but for the sake of brevity, we'll primarily focus on these first two. I mentioned already that we have small molecule antioxidants, so we have ascorbic acid is very important. Ascorbic acid we'll discuss in detail as well. Vitamin E, or the one that vitamin E can refer to the tocopherols, but the one that we're usually most talking about due to its abundance in nature and due to its efficacy as an antioxidant is alpha tocopherol. That's clearly the most important one. You also have things like uric acid, glutathione. We'll talk a little bit about glutathione. Coenzyme Q is, in a sense, similar to the vitamin E species. Beta carotene, we'll talk about carotenoids a little bit, the class that this molecule comes from. And then, of course, melanine is also, you can consider it a free radical scavenger. So these are protein structures, one on the left and one on the right, of two types of superoxide dismutases that are found in humans. And you can see that their structure is very different. If you remember from a number of chapters ago, we discussed how these representations showed you whether you had beta sheets, which are indicated here in this molecule, and then you have random coil structure, which is along this sort of thin line here. If you look at this one, which is called copper zinc superoxide dismutase, it's called copper zinc because it has these copper and zinc atoms that are basically at the core of the protein or the enzyme. And these are actually what give it its functionality as an antioxidant. Basically, reactions, whenever you have enzymes, enzymes usually have some type of active group on it, or sometimes referred to as prosthetic group. And, it could be something as simple as just electrons are passed between this sort of active site and to another molecule. And that's the case with the copper zinc superoxide dismutase that's here. Operating by a similar mechanism, you have what's known as manganese superoxide dismutase, where it has two manganese ions that are present in the protein structure. You can see that this protein has a lot more alpha helical structure than the one on the left. The one on the left doesn't really have any. And then this is accompanied by some random coil structure that's shown, and even there is some beta sheet structure in this polymer, or in this protein. Essentially, both of these enzymes carry out the reaction that's shown here. So they act as a catalyst for this reaction, where you have, if you remember from the last chapter, we talked about superoxide dismutase, and you essentially have two superoxide dismutases here, two superoxide anions that are shown here. These are essentially neutralized in this reaction to hydrogen peroxide and oxygen. So that's essentially the job that these enzymes do in the body, and in this case particularly in skin. So then, the next question that you might ask is, well, now you have hydrogen peroxide present, so isn't that also a reactive oxygen species? And yes, you are correct, it is. And what happens with that is we have another enzyme that's known as catalase. This is just showing you one subunit of the catalase protein. You can see you have a variety of different structures here, random coil, alpha helical structure throughout. You have some beta sheet structure here in the middle. And then what's also important is that you have an NADPH cofactor that's here. You also have a heme cofactor that's also here. And these are the active groups, or the cofactors as they're commonly referred to as well. And what these do is they convert 2 molecules of hydrogen peroxide to 2 molecules of water plus oxygen. So this is pretty good. This basically neutralizes hydrogen peroxide that's present in the cell, and that's the key function of the catalase enzyme. Okay, so now let's move to the small molecules. And when we talk about vitamin C, we're mostly referring to L-ascorbic acid, and sometimes you'll hear it referred to as L-ascorbate, which just means that you have, instead of the a protonated group here, you would have O minus, but basically that's pH dependent. But we tend to think of vitamin C when we think of oranges and sort of citrus fruits, because they tend to be rich in this vitamin. And an interesting thing is that not some animals are actually capable of synthesizing vitamin C, but humans, which share this in common with several other species, the ape, the guinea pig, the trout, are not able to synthesize vitamin C. And it's believed that this is an evolutionary defect, because there's one enzyme that we're missing in the vitamin C synthesis procedure. So there's a good chance that it's a mistake that we're not able to synthesize vitamin C. And that would be a testament of its importance in basically in good health. And you've probably studied things from Linus Pauling in your past, and I don't know if you've ever heard the story about Linus Pauling, but he was a big proponent of vitamin C intake. He was eating large amounts of citrus fruits every day, and he did live to be about, I think, in his early 90s. So whether this had an effect or not, we don't really know that, but there is a good chance that he was on to something in terms of his understanding of the vitamin C biosynthetic pathway, and the fact that we were missing just one enzyme to be able to synthesize it. If you look at goats, for example, like many other animals, they make their own vitamin C, and a goat can actually make 13,000 milligrams of vitamin C per day, and that's just a normal day for the goat. But it can actually make up to about 100,000 milligrams if it's faced with a life-threatening disease, trauma, or stress. And just to give you an idea, to sort of give you a comparison, if you look at the U.S.-Canadian dietary reference intake, they recommend about 90 milligrams per day, and no more than 2 grams per day, which is about 2,000 milligrams. So, the goat requires quite a bit of vitamin C just in its normal daily activities. So this shows you a little bit how L-ascorbic acid works. You basically start with this structure shown on the left here. You can see you have two hydroxyls coming off the main ring. This is where we see the activity occurring primarily. And so, it would basically lose a proton. I think this is actually backwards at the top. The minus H should go with the arrow going in this direction. You essentially lose the proton from here, and you end up with this O minus structure. And then in the next step, you can actually lose another proton, or you could say another electron. And at this point, you would lose it on this OH group that was over here. What this causes is delocalization of an electron in the ring structure. You can see this basically shows you delocalization of the electron, these little dotted lines here going along. So it has like a pseudo double bond character in all these positions. And then a further step would be to lose another electron, and at this point you would basically form these ketone type groups at the bottom here. This is known as DHA or dihydroascorbate. You can see the key for the other ones. You have semi-dihydroascorbate, and then you also have ascorbate, which is shown here. And then of course this is ascorbic acid. So there you can see that vitamin C can actually give up 2 protons. So it can where is it going to give those protons up to, or who is it going to give them up to? It's going to give them up to free radicals. So free radicals that are formed on lipids, proteins, or wherever it may be. It's going to give 2. So one molecule can give up 2 protons, so it can neutralize 2 free radical species. So that's really important. One molecule of ascorbic acid can neutralize 2 free radical species. Here we have, just to give you an idea of vitamin E, and this is olive oil that's shown here on the left, and it's just shown because olive oil is a rich source in vitamin E. And essentially, vitamin E is usually broken down into 2 classes, the tocopherols and the tocotrienols. Basically, they have very similar structure. The main difference in their structure is around the ring here. You can see that they both have, this is known as a phytyl chain. It's PHYT YL. And it's essentially, you can see there's 3 of these repeating units here. So it's a really long chain, and that chain lets this molecule embed in lipid membranes. So before we go on with the vitamin E species, just to set everything straight, vitamin C is water soluble. So one thing about vitamin C is, or ascorbic acid, is that when you intake it or you consume it, it's water soluble. So it doesn't remain stored in lipid tissue or anything like that. So you basically pass it through your urine after a day or something like that. So it's not something that accumulates in the body. Vitamin E, on the other hand, is different, and it does accumulate in the tissue. So you'll have it basically accumulating in lipid type materials. So that's often why they say that vitamin E you need to have on a more regular basis, because you don't have stores of it present in the body. Okay. So when we talk about the tocotrienols, the difference between them and the tocopherols, you can see right away that you basically have a double bond here in this long extended chain. And that's the main difference. Otherwise, the tocopherols and the tocotrienols have the same structure, and you can have 4 different types, that, at least that we're going to talk about here, 4 different isomers. You can have alpha tocopherol or alpha tocotrienol. You can have beta tocopherol or beta tocotrienol, and you can have gamma tocopherol or gamma tocotrienol, and you can also have delta tocopherol and delta tocotrienol. And essentially you can see there that you have the alpha form for both tocopherol and tocotrienol when you have R1 and R2 equal to CH3. So when you have a CH3 group here, CH3 group here, you have alpha tocopherol, and similarly you have, when you have CH3 here, CH3 here, you have alpha tocotrienol. The beta form would be, for each case, when you have R1 with a methyl group or a CH3 group, and then R2, this would just be a proton. The gamma case would be when you have R1 as an H group, or a proton, or a hydrogen atom, and when you have R2 with a methyl group. Same down here. H, or hydrogen in the R1 position, methyl group in the R2 position, gives you the gamma tocotrienol form. And then the delta form is when you have hydrogen in both the R1 and R2 positions for both tocopherols, and also here the same for tocotrienols. So what's interesting is that alpha tocopherol is the most efficacious of all of these isomers of vitamin E, and alpha tocotrienol is also pretty efficacious as well. But the key is understanding why the alpha form is the most efficacious relative to beta, gamma, and delta. And, you know, it all has to do with this OH group. This is where all of the activity lies in terms of the antioxidant activity. And it's essentially, you have this hydrogen atom is donated to a free radical species, and this oxygen-hydrogen bond, its strength is key in this process. And the more groups that you have, like methyl groups that you have here, the methyl groups actually sort of destabilize this ring that's shown here, and essentially pull energy from this oxygen-hydrogen bond and make it weaker. So you want it to be strong enough that it maintains, like it stays there, but you don't want it to be so strong that this molecule doesn't give up the hydrogen atom very readily. So, you know, again, when you have the methyl groups in the R1 and R2 positions, this sort of funnels by induction electrons or electron energy into the ring structure. This sort of causes a destabilization of this OH bond. The OH bond becomes slightly weaker when you compare it to the beta form, the gamma form, and the delta form. The delta form, of course, would be the least efficacious relative to alpha tocopherol, because you would just have protons here, so you wouldn't have electron induction taking place. So the OH bond would be the strongest in the delta form and the weakest in the alpha form. So the alpha form, because it's the weakest, it's able to give up that hydrogen atom most easily or most readily to free radical species. Same basically occurs with the tocotrienols. The same if you go down through. The alpha form would be the most efficacious because this bond is the weakest, and the delta form would be the least efficacious because this OH bond would be the strongest. So here's how alpha tocopherol works as an example. So, it typically is a good inhibitor of lipid peroxidation. Lipid peroxidation refers to, essentially, it's the same as lipid oxidation. They usually refer to it as peroxidation, but it means the same thing. And what you have here is the alpha tocopherol antioxidant. You would have ROO dot, which that is a free radical species. The R is just meant to be the rest of a molecule. In this case, this would be a lipid molecule that has this OO dot species at some point in its structure. And if you recall from the last chapter, we talked about reactive oxygen species, and this was one of the things, I think we've referred to this as the alkoxyl radical. But alpha tocopherol can actually neutralize this by donating this proton that's here to this ROO dot species to give you ROOH. What happens then is alpha tocopherol is left with a free radical species here, so it itself becomes a free radical. But then, just like vitamin C, which can neutralize, one molecule of alpha tocopherol can neutralize two free radical species or two reactive oxygen species. You have another free radical that's here, and alpha tocopherol can then donate another proton to that, and it results basically in a transformation of the molecule where you have this sort of quinone type structure. You have two ketone functional group here and one over here. You can see that this second ring here opens up. You still have the chain that's going along, and you have basically a molecule known as alpha tocopherol quinone now. So again, one molecule of alpha tocopherol neutralizes two free radical species. The carotenoids. The carotenoids have a very distinct structure, and we think of a lot of different vegetables when we think of carotenoids. Beta carotene, we always think of carrots, have that sort of distinct orangish color that comes from beta carotene. Spinach is rich in a molecule known as lutein. This is another carotenoid. Tomatoes are rich in lycopene, which is another carotenoid. And from a molecular structure standpoint, you can see that beta carotene and lutein have a pretty similar structure. Basically, you have this series of basically a series of conjugation that occurs here along this alkyl chain, and then you sort of have these end groups, these cyclic end groups for both of these molecules, but you don't necessarily have to have the cyclic end groups to be in the carotenoid class. You can see lycopenes here have this series of double bonds present in its structure as well. And the way that these work is if you have a free radical species, again, we're using the example of a lipid where you have, we're referring again, ROO dot. R would be the rest of the lipid molecule. You would have this OO dot species at some point. And the way that beta carotene works is that it actually sort of acts as a radical sink where you have that conjugated structure that's shown here. And it basically traps the radical along this conjugated structure. So one difference that you can see here is that, and here you can see the ROO group basically becomes bound to the carotene molecule. So, again, you're not repairing anything with these antioxidants, but you are stopping the propagation of the free radical mechanism that's taking place. Again, this is like a free radical trapping mechanism. The radical becomes trapped in this, along this conjugated bond structure that's in the molecule. And from that point on, you really don't, you only have one beta carotene that's able to neutralize one free radical species. So that's a little bit different from what we see with the vitamin E species, and or at least alpha tocopherol, and also with L-ascorbic acid. Glutathione is another really important antioxidant in skin. And again, I'm saying in skin, these are also important in other parts of the body as well. But again, we're most concerned with their presence in skin. This is the glutathione molecule that's shown here on the left. It's composed of this gamma-glutamic acid structure that's shown up at the top here. After that you have, so this is like a tripeptide. You have a cysteine molecule here, and then you have a glycine molecule here. And again, glycine, you'll remember, the R group is H, or a hydrogen, so you have CH2. Cysteine is this sulfur-containing R group, which is shown here. And then glutamic acid is actually where you see this part of the molecule. This is the R group coming down here, CH2, CH2, and then you have C double bond O, and this normally would be in just glutamic acid, the amino acid. This would be COO, C double bond O, and then an OH, but in this case it's bound sort of like it's bound to this other amino acid, so that you basically allowing it to join cysteine and glycine together. So glutathione, you have shown here. This is showing you in its regular state, what it looks like. When it becomes oxidized, basically you form, if you remember when we talked about hair, when you oxidize, oxidation results in the formation of disulfide bonds. Reduction, which would be going back this way, results in the breaking of disulfide bonds. So the oxidized form looks like this, and you also have water that's formed or evolved from this reaction, and then you end up with this ROH species that's shown here. So this would be starting with an ROOH that you show here, which is basically not a good, but also a damaged species, a reactive oxygen species, and glutathione takes care of it in this manner. Glutathione, we'll talk, ascorbic acid and glutathione are really important because they can regenerate alpha tocopherol. And this sort of shows it diagrammatically how this works. If you could imagine that this portion of the slide that you see now on the left is the membrane area, and the portion on the right is the cytoplasm. So this is aqueous on the right, this is lipid soluble on the left. So we know that alpha tocopherol is a lipid soluble antioxidant, so it resides on the left here in the lipid bilayers. So here we show alpha tocopherol basically neutralizing, this ROO dot or ROO free radical species, forming ROOH, and then you have the formation of alpha tocopherol radical. So you can have glutathione basically interact right at the membrane, at the interface of the membrane with the cytoplasm. Basically, it could regenerate alpha tocopherol, but then you end up forming the glutathione radical that's shown here. But it's showing you, lipid peroxidation is a really important process to prevent, and essentially this water-soluble antioxidant is interacting at the interface, sacrificing itself to reform normal alpha tocopherol, regenerated alpha tocopherol. Similarly, you can see below here we have ROO dot being neutralized by alpha tocopherol, forming ROOH, which again, ROOH is a structure that's, it's not a functional structure, but at least it's a neutralized structure, and it stops the free radical events. So alpha tocopherol radical is formed as a result of this process. And then you could have ascorbic acid or ascorbate essentially react at the interface with alpha tocopherol, the alpha tocopherol radical, to regenerate alpha tocopherol. This results in the formation of dehydroascorbate, which you saw on the previous slides. So this is sort of like a radical form of the L-ascorbic acid molecule. Glutathione can actually interact with this and help you reproduce the ascorbate, the ascorbic acid that you started with. So then ascorbic acid could go on to regenerate another alpha tocopherol molecule, or it could interact and neutralize another type of reactive oxygen species or free radical species. So that brings us to the end of that chapter. There's a lot more that you could read about in the textbook. If you do, I would recommend reading the sections that are relevant to what we covered here in the class. There's some interesting topics in the text that are covered by that. I would pretty much focus on, the items that we discussed here, and see what you can read in addition to that in the textbook. If you have any questions, please contact me via email, and I'll try to get back to you as soon as possible. So I hope that you enjoyed this introduction to the endogenous antioxidants in the skin, and we'll pick up next time, I think, with looking at solar radiation of the skin and how it influences the skin. Thank you, and goodbye.